Hello, everyone. I hope you can see my screen.
Yes.
Thank you very much for the invitation to participate in the SymbNET program by sharing our expertise actually in small molecule metabolite analysis. So I'm leading the Metabolomics Service and Research Platform at University of Lausanne. And it's my pleasure to walk you through actually our activity. So I will do this by presenting you our mission, the composition of our team. I will go through our service catalog, and then also at the end, I will provide you a brief overview of our own actually research and development projects. So our platform was created actually back in 2016. So it's we are a very recent facility with the aim really to provide the advisory, analytical, so experimental, and then also bioinformatics support to the research groups that are interested in performing the state-of-the-art mass spectrometry-based qualitative and quantitative analysis of polar metabolome and lipidome. So basically the small molecule complement of cellular metabolism. In addition to these, really to the service activity, we also have our own research and development goals. So the first one is really to implement new methodologies with a focus on quantitative metabolite analysis. And the second one really to apply these methodologies from model systems to human population studies to in general enhance our understanding of metabolic alterations with aging. So we are very well equipped for efficient sample preparation, also, of course, data acquisition and data processing and analysis. And we are today collaborating with more than 60 different research groups across Switzerland and beyond. So our overall goal is really to facilitate the study of metabolism from the metabolite perspective and to enhance, hopefully, the understanding of metabolite roles and activities at the systems level in health, in aging, and in disease. And with this scheme below, I just wanted to illustrate actually this how metabolomics today, due to mainly its strong phenotyping potential, but also relatively high throughput, is actually already widely applied from model systems to human population studies. So we can today really analyze actually thousands of samples in the context of human population studies as well. Our team is composed of the experienced staff involved in the service activity. And then also we are hosting on a regular basis students and trainees who are working on our own research projects. So in terms of technologies, we are equipped with 5 different LC-MS systems, out of which 2 are high-resolution mass spectrometers and 3 of them are tandem mass spectrometers. We have recently also installed one GC-MS system and as well the robots for liquid handling to really enhance actually the reproducibility of the sample preparation. In terms of approaches and analysis we can perform. So I have just illustrated. I mean, I put you here are summarized. So all different, let's say, our service catalog in this table. So we are performing untargeted analysis. This can be done either with a focus on polar metabolites or with a focus on complex lipids. This type of analysis is of course considered as a discovery approach without an a priori hypothesis, and it's based on relative quantification. We are also performing the stable isotope-assisted analysis to track the fate of labeled metabolites. We have developed several methods to perform actually I'd say, to target as many metabolites as possible. These methods were actually developed as an alternative for the untargeted analysis. And today, we are really proud that we have set up actually two methods, one for multiple pathway targeted of complex lipids. And with this method, we can actually actually quantify more than 1,000 different lipid species belonging to 5 to 6 different lipid classes. We have a similar method developed for polar metabolites, with which we can also quantify more than 500 polar metabolites involved in central carbon metabolism. We have also set up many different methods for a single pathway targeted analysis with a focus on specific class of metabolites of interest or specific pathways. And these include the amino acids, bile acids, steroids, acyl carnitine, sphingolipids, eicosanoids. I mean, you can really see the entire catalog on our web page. So don't hesitate, to contact me if you need more information about this. Just to give you the idea about the sample types that we can actually analyze, there are no limits in terms of different biological matrices that we can analyze. But in most of the cases, in terms of if we take a look at the statistics, based on the last 3 years of since we were fully functional, we are mainly analyzing different types of biofluids, so including plasma, serum, urine, CSF, and then of course a lot of different tissue extracts, cell lysates, including of course bacterial pellets, yeast extracts, etc. We can also analyze many different alternative, more exotic samples such as tears, sweat, hemolymph, fat bodies, fish mucus, different tissues from different species of marine invertebrates, plants, etc. So really, there are no limits in terms of sample types. In terms of the research and development, I will now just briefly present you our projects covering 3 different aspects. So the first one concerns the analytical method development. The second one focuses more on development of tools and databases that we are developing actually to facilitate the data analysis and interpretation. And the last one actually involves several different research projects that we are involved in with clinicians and data scientists here in Lausanne. So in terms of analytical method development, I have already mentioned this, but we are really mainly focusing on the development of high coverage targeted methods, mainly to bridge the gap actually between 2 existing extremes. So on one side, the targeted methods, the classical targeted methods with which we would rather focus on one specific pathway or one class of metabolites of interest. And on the other extreme, actually the untargeted analysis with which we are always trying to measure as many metabolites as possible. So as an alternative for this untargeted analysis, we are actually developing the high coverage targeted methods with which we can really today easily measure several hundred polar metabolites in a single analysis, and also up to 1,000 different lipid species. In terms of data analysis and interpretation, we are currently designing and implementing a Metabolom Explorer, so an interactive database to store and explore the metabolic signatures and the quantitative data on the metabolites that have been measured in different biological matrices. So this is an ongoing project in collaboration with the Bioinformatics Competence Center here at University of Lausanne. And then finally, we are also involved in several projects that are really embedded in a specific context. So we are really exploring, and looking to characterize actually the Alzheimer's disease as an energy metabolism disorder. And this is at the moment done at several different levels, from skin fibroblast analysis to the analysis of mouse brain mouse tissue, and then CSF and plasma analysis in the context of human population. So we are working on on two specific populations, patient populations here in Lausanne, and in collaboration with clinicians from the hospital in Grenoble. On the top of this, we are also involved in two big population studies, so here in Lausanne and in Bâle, so the Colaus and the Complete Health and the Heart study. These are really large-scale population studies, prospective studies, so longitudinal studies, with which we are actually trying to understand how the metabolic signatures can help us to predict the health outcomes, and then hopefully serve as a tool to prevent actually the onset and progression of non-communicable chronic metabolic diseases. And with this, I would like to thank you for your attention, and yes, many thanks to Philip Engel, who has actually invited us to participate to SymbNET.
Thank you very much, Julijana. Maybe just a quick technical question. Does the metabolite extraction work equally well for tissues as cultured cells, or are there any technical challenges between these two?
No, not really. We don't see, we use the tissue homogenizer. So any kind of tissue actually can be easily homogenized with the use of beads, of ceramic beads. So yes, this is quite a commonly applied protocol, and no, we have not really encountered any issues, no matter if it's whatever kind of tissue it is. Yeah.
All right. Thank you.
Then it's of course the question of normalization at the end. You have to think about how you will normalize. Whether you will, the metabolite concentrations can be reported to the protein content, for example. This is what we are doing in most of the cases, or then to the tissue weight. But if you use the tissue weight, you can introduce a significant bias. So yeah, you have to think carefully about the normalization, which normalization technique you will apply.
Then there's a question, what about feces? Is it harder? I guess, whether it's harder to extract metabolite,
we are actually, in the context of the Lausanne population, we are starting now to analyze feces samples. Feces is usually lyophilized prior to metabolite extraction. And then we are reporting actually the metabolite concentrations to the dry bath. This is actually, there is an intention to actually correlate the gut microbiome diversity to the metabolic signatures of feces, yes, in the context of Colaus.
All right, and then there's a question from Luis, so let's maybe have one quick question and then we proceed.
Just I don't know if I understood correctly. So do you have a database of metabolites for different model organisms? That's what you're doing?
We are actually now developing this database, yes, because we are really uniquely positioned, if you wish, to because we are analyzing so many diverse sample types, and we have set up a lot of quantitative methods. So we would really like to now record all of this data we have acquired in a very comprehensive database that, yes, we'd like to make publicly accessible.
And one is Drosophila, right?
I'm sorry?
And one is Drosophila melanogaster?
Drosophila will be among the organisms, yes, correct, because we have analyzed a lot of brain tissue and fat bodies from Drosophila, yes, including the hemolymph as well, yes.